Hello everyone, I'm going to present my work about comparing cortical plasticity induced by conventional and high-definition TRNS. Since it has been found that motor cortex stimulation can induce limb movements in 1870s, scientists are making use of brain stimulation, and there is a new wave of brain stimulation method called transcranial electrical stimulation. This method consists of transcranial direct current stimulation, transcranial alternating current stimulation, and transcranial random noise stimulation. These stimulation methods are non-invasive brain stimulation techniques which are being used extensively for neuromodulatory purposes for various domains. There are three important parameters when applying transcranial electrical stimulation: current flow direction, amplitude, and electrode montage. By the way, transcranial means through the scalp. Okay. In conventional ways of transcranial electrical stimulation, you need to have at least one stimulating electrode. You can have more than one. One stimulating electrode and one reference electrode. It has been shown in the neuroimaging studies by Lang et al. 2005. It has been shown that if you stimulate with these conventional parameters, you will end up with larger area, affected larger area than as you wish. And in 2007, Nietzsche et al. Showed that if you reduce the size of the stimulating electrode and increase the size of the reference electrode, you will end up with more focal effect. And by these ideas, there is a new line of research opened. It is called HDTDCS, and this consists of 4 return electrodes and 1 stimulating electrode. And this method has been shown to be more focal with both computational modeling and experimental studies. So TRNS, what is TRNS? Transcranial random noise stimulation is a special form of alternating current stimulation which is involving stochastic randomized fashion from 0 to 640 Hertz. And this was first implemented by Trnas et al. In 2008. And there's 2 forms of TRNS you can apply. Low frequency TRNS is 0 to 100 Hertz, and high frequency TRNS is low frequency TRNS from 100 to 640 Hertz. And there is 2 suggested mechanisms of actions for TRNS. One is Paulus proposes that it might arise because of the ongoing openings of the sodium channels, so hinting membrane depolarization. But the other suggested mechanism is that stochastic resonance. And in this phenomenon, the random noise stimulation increases the sensitivity of the neuronal population, so that the firing threshold could be reached more easily by Fertonen et al. Okay, here we see the results of Trnas et al. This was the first paper on TRNS, and it helped a lot to understand what is TRNS actually. So here we see the cortical excitability fashion for high frequency TRNS. Here we see the low frequency, and here we see the sharp stimulation. As you can see, high frequency can increase the motor cortical excitability up to 50%, but low frequency doesn't do anything. So by this idea, we used low frequency as our control condition, and we want to compare HD TRNS, conventional TRNS, and low frequency TRNS. Of notice, this is the first study ever implemented for HD TRNS. We would like to, there are some studies with HD TDCS, HD TACS, but not for HD TRNS. This is the first study ever been implemented. So our study design, we have 2 blocks of TMS. We get 2 blocks of TMS baselines, and then we have 10 minutes of random noise application, randomized condition HD, high frequency or low frequency, and we have 7 blocks of after effects of the random noise stimulation. So as I said, we have 3 sessions in total, at least 3 days in between. For HD we have C3 as a stimulating electrode, FC1, FC5, CP1 and CP5 are return electrodes. For the other stimulation protocol, we have C3 as again the stimulating electrode, and the reference is put on each lateral shoulder. And we get the subjects with individual MRI scanner to navigate to stimulate exactly the area. And we have right-handed subjects without neurological and psychiatric disorders. So continuing on study design, we have for HD we have intensity 0.5 milliamp, and the size of the electrode is 3.14 centimeters square, and the density is calculated accordingly. And for other protocols we have again the same density, but the electrode sizes are changing. This is the 3.14, this is the 8 centimeters square, this is 25 centimeters square electrodes that we use. And the density is calculated according to density is just intensity over the area of the electrode. So we used 8-shaped coil to stimulate, and when we conduct the experiment, we first find the hotspot which is producing reliable MEPs, and MEPs were recorded from the right first dorsal interosus muscle, and we first find the motor thresholds, and then we stimulate 110% of these motor thresholds. As I said, we record 2 blocks of baseline and 10 minutes of TRNS and 7 blocks of after effects with 5 minutes break, and every block involving 20 TMS pulses. Here we also made use of a calculational program embedded into a neuroelectric system to foresee the actions of the brain stimulation beforehand. So here here we can see conventional is highly distributed, but high definition is looking like more focal. And here is our hypothesis. As I say, we want to introduce a new novel methods, HD TRNS. It was never implemented before this study, and we want to confirm this, the results of Trnas et al. For conventional paradigm, and we want to examine the HD transcranial electric stimulation methods if it is more focal and the efficiency of HD TRNS on motor cortex plasticity. Here we can see the results of the of the experiment. Here we have 3 groups. This is the conventional high frequency group, this is the low frequency group, and this is the HD stimulation group. And here we can see we don't have significant effects here, but we did repeated measurements of ANOVA with 3 by 7 conditions, conventional HD and low, after 1 till after 7, and to see the effects. And ANOVA showed a non-significant for different stimulation protocols. It was like 0.1, but we were close to significant for time points, and interaction between time and TRNS was not significant. Also, though we are not significant, there is a trend here. You can see this line corresponds to a conventional high frequency, and it is nearly 40% of it is nearly increasing 40% the motor cortex excitability as compared to low frequency, and low frequency maintains it doesn't change so much. So this is our control condition, and for HD we have up to 30% inhibition. So also we collapse all the data here. For example, like for conventional here, we collapse all the data to see the total effect. And here the total effect also confirms that there is an increased excitability for conventional, but decreased excitability for HD. This is all. Thank you very much for listening. If you have any questions, I can answer them.